Just starting off here, my background information is going to have been covered by several people thus far already too, so I'm going to keep it relatively short. One thing that I have spent the last about 10 months doing is putting together a perspective article, looking at several of the different herbaceous species and their particular responses to changing climates. Looking all of these data through things like bucera and things like monkeyflowers, Ipomopsis, Mengus cardinalis, partridge pea, and a couple of different grasses as well too, switchgrass out there. And I want to say that looking at this, there's a couple of things that just pop right out at you. First of all, almost every one of these shows maladaptive or declining populations in at least part of their range. They show species distribution models that predict declines in parts of their range as well. They show that plasticity isn't sufficient to actually match future climate projections, and they often show that species are dispersal limited and are not going to make it to different areas in time. So having said that, we need some management strategies that may account for some of this. And assisted gene flow is one of these answers or options for this, and we're simply translocating individuals within their species range, either with the purpose of facilitating adaptation by moving areas from, say, hotter and drier climates into other climates where they now match the current climate that's there. And this can also just increase genetic diversity in the target populations for any populations that have any climate variety. So let's create some predictions really quickly about what would happen if we do assisted gene flow. So relative thickness on the Y, time in generations on the X, AGF is a line right here. So from our baseline data saying that we have relative fitness around 1 starting, if we have some populations where we do assisted gene flow and others that are control, what you might see is that in future climates, these control populations, you wouldn't be moving individuals into them unless they were declining, may be declining rapidly, whereas your AGF population, the hope, right, is that you maintain higher fitness than in those control populations where you did not move those translocated individuals. You can visualize this as a box plot easily too, with the yellow bars being baseline data before assisted gene flow, and the green bars over here being the generation 4. In the control, you might not see any difference between the two generations if climate conditions have not changed. But in the AGF population, you would hope that you'd see some elevation in fitness in those populations. We just moved a bunch of individuals from far away into there. So you also can think about, besides the mean, that you probably are going to see an increase in variation in fitness within those populations as well. Now we all know here probably that assisted gene flow is a controversial management practice right now too. So we should talk about why it's controversial and what the actual questions are that we need to answer to make it either viable or not viable, right? So this is the general model you have for assisted gene flow. We have population growth rates, you have a declining population here already, you're going to introduce individuals, and almost all models predict that are going to see some sort of maladaptation at the beginning of this specific hump. So how deep that trough goes is a question, and then you're going to get to the evolutionary rescue side, but hopefully you're recovering some level of land up to be above 1 for your population. Now, we don't have a lot of examples of this in natural populations. What we have is we have laboratory examples in yeast and several bacteria, and we have a bunch of examples with relatively poor experimental designs. And that's because this has been done in threatened species, right? We're doing this in giant sequoias after gigantic fires. We're doing this in coral that has been completely bleached out. And we're doing this in partridge pea, which is a better example and has a better experimental design, but not really done in great. Currently happening right now. Okay. So we have questions that come out of assisted gene flow as well too. So what if those native distant hybrids are maladaptive? That Alpine depression that we've talked about already. What if the distant alleles completely swamp out the native alleles in those sites and lose genetic diversity? And what if those distant alleles are only favored in some years, but they're not favored in other years? We really don't have systems where we've answered these questions to the fullest extent possible. So let's bring in Mengus cutaneous. This is not a threatened species throughout the vast majority of its species range, okay? It has a wide geographic range. It occurs all the way from central Mexico to central Alaska, from sea level to about 11,000 feet above sea level. It occurs at 11,000 feet as an annual, that's about that big. It occurs on the coast, as you know, a creeping, stolen thing I could get from my waist. So really diverse phenotypically, and one of the most genetically diverse species that we have seen. Okay, it's a wonderful ecological model system, dates all the way back to Clausen-Kekep-Heise for its California ecology. But it also has a lot of nice molecular tools associated with it as well, including a relatively simple genome and many resequenced genomes out there. What you need to know for this talk is there are some areas within its range that it is threatened by changing climates. These are the areas that I work in. Okay, what's the evidence for that? So one of the experiments I did about a decade ago now, in 2014, we had moved populations from low and high elevation California to low and high elevation Oregon, and we did a common garden study in low and high elevation in Oregon. And we see the signature of an adaptation lag in both the low and the high elevation populations in California. This was not expected. Muscatatus has really high levels of genetic variation. We thought it would have been adapting to these changing conditions. So in the low elevation garden site, you see that the California low elevation populations do much better than the Oregon low elevation populations. And the same thing happens in the high elevation site. The California high elevation plants do much better than the Oregon high elevation plants in those two gardens. This was only a single year, but we've done this experiment again with more populations. Not as many as Jill, for not as many years as Jill, but still always consistent with an adaptation like here. And we think for these populations, this might be kind of canary in the coal mine. If this species cannot adapt, pretty much no species can be able to adapt fast enough to keep up with changing conditions. So I started a faculty position about 7 years ago, and I said, I want to stay in these subalpine meadows for my entire career. So I started these long-term observational experiments, looking at 12 different populations in the Central Oregon Cascades, at high elevation sites because they're easier. Every year we go out to these 12 populations. We have a couple transects across them. We collect seeds from about 50 individuals, a number of flowers, and a couple of simple phenotypes in the field. We bring those seeds back to the greenhouse. We plant them. We conduct a common garden on them. We look at a bunch of different phenotypes, morphological, phenological, physiological. And then we take tissue from those plants and sequence whole genomes of 24 plants per individual per year. We've now done that for 7 straight years. And what we found in that is that one of these populations, when extirpated for a couple of years, then popped on, but it is slowly spiraling towards extinction, and that a couple of the other populations are not looking great either. So 3 years into that, we decided that this would be a perfect opportunity to use this for an assisted gene flow experiment. In the first years of this, and everything I want to present today is with my master's student, Donna Hendricks, who just graduated recently. So a lot of this is her models and her figures that you're looking at. What we did was we took the two populations that had high fitness and alternative years. We created F1s out of those two populations, and then we planted those F1s in landscape level experimental design. So for those 12 populations, we had 4 sets of 3 populations. In those 3 populations, we had a control group where we threw out 200 native seeds, a seeds group where we threw out 200 of those F1 seeds from California, and then we put in seedlings, about 20 seedlings, to get rid of that germination step and make sure that we overcame that step in these particular populations. So we have 4 replicates of those 3 kind of populations that we can track alleles in. We have 3 years of baseline data, and then I'm going to talk about 3 years after that. So this is a really early period. Assisted gene flow probably doesn't work this fast most of the time. All right. Cool. So what do we actually see here? So if we look at floral abundance on this side, what we see is in the seeds treatment, we see an increase relative to the baseline years. And in the control treatment, right, you see relatively similar means in that control treatment. That is, we see about a 20% increase in floral abundance in the populations that we seeded with those California alleles. We see no increase really in the seedlings population. In terms of variation that you see, we see what we would expect here. In that seeds treatment, we see higher levels of variation in that seeds treatment than we do in the control treatment relative to the baseline group for floral abundance. Now, this translates only marginally to seed production. So you're producing more flowers, but there's not necessarily more seeds in these different populations. So again, we see about a 20% increase in the seeds treatment relative to what's happening in the control treatment, and we see less happening in the variation there. So, I'm telling you here that assisted gene flow was effective somewhat in the seeded populations. We're at least producing more flowers in those populations, but it looks really variable in how it's actually happening. And we're wondering, why didn't it work in the seedling populations where we know we actually put seedlings in? Well, it's pretty simple. The year we planted it out, there was this gigantic heat dome in the Pacific Northwest this year, where the temperatures in Portland and Seattle got up to 115 degrees Fahrenheit. They also peaked very high, almost 100 degrees at 4,000 feet in the Cascades as well too. This is before, this is a couple of days after. This is our seedlings before. This is our seedlings a couple of days after. So they didn't do very well for those seedlings that year. But the seeds didn't all germinate that year, and we think that's why it was more effective in the seeded treatments. So these seedlings did produce some flowers, but just not that many, and we're still wondering if there's going to be some introgression of those alleles in those populations. Okay, so did all populations respond the same way across all 3 years? And the answer is of course not. Like, that would be too easy, right? If that's like what you wanted for assisted gene flow here. So one of the reasons you see that much variation occurring in the floral abundance treatment and the seeded treatment is because there's variation among those 4 different population groups that I talked about before. Some behaved, as you may expect, for floral abundance, where the control population over here, we actually see lower relative fitness relative to the baseline years, but the seedling population and the seeded population both show increases in floral abundance, right? Other ones, we see, decreases in the control population, but maintaining essentially the baseline. These are the lowest elevation populations in our group. This is the highest set of elevation populations here. But there are others where we see no introgression in either the seeds or the seedlings treatment, where clearly nothing has happened very well in those treatments. We also see heterogeneity between the years that we do this. For instance, in 2022 when we do this, we see that there is higher fitness in that year than all of the other years, and we see that there's more an advantage of having seeds in that year as well. In 2023, it was a far worse year. We still see an advantage in the seeds versus the control treatment, but it's much less pronounced in those particular years too. So there's definitely heterogeneity between years going on here too, that we need to think about when we're thinking about populations that we're planting in, and how much genetic diversity we're planting in these populations. So the first question I was going to ask is, how sure are you that AGF is actually causing those fitness changes there? So we decided to validate this using our genomic data to see if we have alleles from California actually inserting into those Oregon populations. This is a pretty standard bioinformatics pipeline. The takeaway is we have about 15 lines per year that had, no matter our final filtering criteria, we have about 11x coverage, so we're able to call genotypes in a reasonable error rate. What we did is we looked for alleles that were private to California. That means these particular alleles at a gene are only found in California, but they aren't found in the baseline populations in Oregon. And then we tried to track those alleles after we had assisted gene flow, right? We expect some introgression of those alleles if assisted gene flow was successful. So we tracked about 159,000 SNPs that are private to California across the genome, and that were at fairly high frequency in California as well too. So we're likely to get them into our populations. And indeed, we see some introgression in different years in these different populations relative to control. This is the control population over here. There are less introgressed California alleles than there are in either the seeds or the seedlings treatment. There are still some, and that's largely to do because of the way we calculated private alleles. But also maybe because our alleles are leaking down the hill to the populations that are 200 meters away as well. What's crazy about this, though, is we see different private alleles introgressing in 2022 versus 2023. And we only see very small parts that have benefits in both years, because these years are very different than one another. So this can be a large problem that we have to start thinking about, too, when we're thinking about assisted gene flow. So just to put this all together right at the end here, we could think about this in a way here for our validation. So if we think assisted gene flow actually works, we should have a benefit in fitness in that population, plus we should show introgression of those alleles, right? That would be the ideal. We could also see maladaptation. So we could get introgression, but it could not benefit in fitness. That might be what you'd expect for the first generations. In either one of these, we don't have integration, so we don't expect that those actually work for assisted gene flow. That's just what I said. Okay, so we see some that are in this upper quadrant up here. One of our 4 site groups is in this quadrant, where compared to the control populations, we certainly see integration, and we certainly see a benefit in fitness, mostly in the seeded populations. Others are in this maladaptive section down here. One of the 4 is in this section, where we see integration, but we see declines actually in fitness in the seeded populations. The 2 other ones, we don't think there was any integration in there, and all the changes that we're seeing are stochastic. Okay, so results. We think we see some rapid success of AGF in some populations, and we think we see that maladaptation kind of hump in other populations. We're on the hook for surveying this for another 6 years, so this is what we'll be doing. The AGF response is definitely heterogeneous across populations and time, which is something we need to think more about for how much temporal variation there is in sites. And integration definitely is occurring genome-wide, and the frequencies are quite low, as we would probably expect from early on introducing a relatively small amount of alleles. So we're also determining kind of the genomic, ecological mechanisms, the traits that are actually causing these differences in fitness, and that will be for next time. But we have some takeaways from management too. So our success is more rapid for that one population than assumed by many of the theoretical models here. The recommended introduction of 5 to 20% of the population, we erred on the low side because I was scared. So we put about 5% of the population. Clearly our seeds work better than our seedlings, and we think that's because they could come up in multiple different years, and we could use dormancy to our favor. That temporal heterogeneity is going to have really significant effects, and we could, by having 1 or 2 bad years, all of our introduced plants may not actually be able to integrate into the population. So we may have to think about doing these introductions more than 1 year to make sure they're actually working. And this has brought up a lot of issues about what are the best practices for actually choosing germplasm. How do we pick the populations? How many populations do we throw out there? Do we do crosses? And there's a lot of different possibilities. Just distant seed, we can put out native distant hybrids, we can put out distant hybrids, or we can just throw a bunch of seed from a bunch of populations and let natural selection do it. I would advocate against that because they'd be at low allele frequencies, and we probably wouldn't get the alleles we want in those populations. Okay. Last, I want to talk about later on at some point, do we move species that are not threatened with extinction because they may be dominant species and they may have effects on species that are threatened with extinction? I think this is something we need to talk about, being proactive rather than reactive. Thanks to everybody that contributed to this, specifically Donna, and specifically the U.S. Forest Service who gave me permission to do this, which was lovely of them, and NSF funded it. I think I'm over time. So.